So, how to get this experimental data? Because we discussed about different types of experimental techniques. What are the different experimental techniques?
Cyclodichroism.
Right, cyclodichroism, different scanning calorimetry, fluorescence spectroscopy. So, we have different experimental techniques, and for each protein, right, they mutate the residue and use the techniques to estimate the free energy change upon mutation. This wild type residue we know, right, for example 10 kilocalorie per mole, and upon mutation if it is 12 kilocalorie per mole, then the difference is 2 kilocalorie per mole. This difference is mainly due to the mutation of a particular residue, right, from the wild type. So, to understand this, right, there are several experimental groups, they are working on different types of proteins. For example, Alan First from the Cambridge, right, he is using Barnes as a source, right. So, using the Barnes protein, so they try to mutate different residues and understand the effect of mutations in the particular protein. How far the stability changes, right, at different positions, whether the mutation is any specific residues or any secondary structures or based on the organ accessibility and so on. So, Nick Pace, right, he is from the University of Texas. So, he is working on ribonuclease T1 and he analyzed the mutations and showed that hydrogen bonds are important factors for the stability of the particular proteins upon mutation. Then Brian Matthews carried out the investigations on T4 lysozyme at 164 residues, and he systematically mutated all the residues, all the 164 residues, right, and then he analyzed the stability upon mutation. He also get the structures of all these mutants and he compared the 3D structure information with the delta G values, and in this case he can be able to tell which mutations involve which type of interactions and specifically how much kilocalorie is required to form any specific type of interactions because of the crystallographic structures as well as the free energy values. Likewise, there are different groups like the Utani is reported for lysozyme, right, David Shortell the strap nucleus and C.R. Matthews on tryptophan synthase. So, earlier days they tried to use some proteins, right, and currently if you see the literature they are mainly focusing on the important proteins of functions or the disease causing mutations. So, you try to get the new proteins and identify the important locations and trying to understand how the stability changes and how we attribute the stability with the different other features. When they started the experiments in earlier years, right, they systematically carried out the mutations to understand the factors influence the stability. The current scenario, right, because we are going very far ahead. So, they want to see any particular mutants which are important for function or important for any diseases, right. So, they are trying to do it. This is the reason if you see the current literature, there are many proteins, we know the mutations, but not many mutations in a particular protein. If you look into this T4 lysozyme or the Barnes or ribonuclease, you will get hundreds of data, but several proteins they have only 1 or 2 mutants, right, it is available in the literature. So, these data they publish in the literature and they are available in the literature. It is very important to collect this information and use it for the application purposes. So, in 1998 Feil is from Germany. So, he collected the data and he published a book on thermodynamic data by Springer. So, even they published a book, this is not accessible to several investigators, right, and it is required to get the data and show this in the form of computer readable form. So, in this case what can you do, it is very important to develop a database, right. Earlier days because of not good computers are available as internet facility was not there. So, they collected the data and they published or they kept the data with themselves for analyzing the data. Currently, right, due to the availability of fast computers and the high storage capacity, it is easily possible you can see the transfer the data. So, it is we can do it, develop a database. In one of the earlier classes we discussed about the development of database, right, what are the various features of database, right, and how to analyze the data from this database, right. So, now in this thermodynamic data for the proteins or the mutants, right, we collect all the data, right, put in a computer readable form, and it is very important to provide several options to search the data, because each user has a different options to search the data because they are interested on different types of data, right. I will explain some of the details in the later part. So, it is important to give the search option. so that they can search for any data they require, and we need to display the results, right, in a specific format where the user requires. Then it is also important to give the data to be downloadable. In that case they can download the data and they can analyze the data. So, it is important one to develop a database for the mutants as well as for the proteins. Once the database is ready, the data are available, then you can use it for the analysis. Either you can analyze the residues which are stabilizing or which are destabilizing, and what are the factors which are important to understand the stability. Are there any relationship between amino acid properties and the stability upon mutation, any specific properties which can influence the stability of the mutations. Once you relate the properties and the stability, then it is possible to develop models. In this case you can predict the stability upon mutation for unknown data. So, these three different aspects. The first one is developing a database that should be available, and you should have the search options, and the result you have to give the option to download. Once a database is ready, then the second aspect, we can analyze the data to understand what are the factors which are important for the stability. Once we analyze the data, we will get the hypothesis, right. These are the various aspects of the bioinformatics, right, as discussed in the first class. Right. Then you can develop models for the prediction. These aspects you can use for different aspects. For example, stability, binding affinity, right, of the protein-protein interactions, protein-DNA interactions, right. For any applications we can use this type of approach. So, how to make a database? What are the important factors we need to consider for developing a database, right. So, first you need to put in the important information what you are interested in. Here I put a thermodynamic data, right, for the proteins and the mutants. So, put the them here, this is the database name. So, mainly we give the data. So, what the experimental data we get? Delta G obtained from thermal denaturation, or you can obtain from the denaturation, because we put H2O, because if you denature the protein we add the denaturant. So, we get the values any particular concentration. If you extrapolate these values to 0 concentration, you will get the value at 0 concentration. This is to have the data uniform, otherwise you have the value for different concentrations. In this case it is not good to compare. So, we extrapolate the value to 0, so we get the value at the 0 concentration. Then you can get the thermal, you can see melting temperature, and the change in melting temperature will get delta Tm we can get. This is experimental data. This experimental data depends upon the conditions, what are the conditions we use, as well as the method, what are different methods we use. We use the CD or DSC or fluorescence we use, and different conditions, for example temperature, pH, different buffers and ions and the concentration. So, the major aspect of the database is the data along with the conditions, this is fine. Now, to facilitate the users, right, for giving more information to the users, we can add the literature where exactly we get the data, right. If any of the users they are interested in particular protein or they want to know more about the data, then they can go to the reference and they can use that reference to understand more about the particular protein on the stability. So, in this case one part is done. Then if you take any particular protein, so you can see it as a particular sequence, there are some particular structures if it is known by the X-ray crystallography or NMR spectroscopy. So, you can give the sequence information. So, where we get the sequence information? Uniprot, right. Earlier days we had two different databases called PIR. What is PIR? Protein Information Resource and Sysprot, and they merged to Uniprot. So, currently we have the Uniprot ID. Then for the structure, PDB, right, this is a unique resource protein data bank. So, we give the PDB ID. Then also we have some motifs and the mutations. For example, if it is mutated, right, we will give the wild type residue and where we mutate and to which residue we mutated. So, we give all the information. So, when you give all the information, this is complete, then we can start this use this database by different search options, and you obtain the results, and then you can use for your analysis and for developing any algorithms for predicting the stability. Now we will explain about the utilities of database and the future classes I will explain about how we can use this database, right, for understanding the factors which influence the stability as well as development of the models. So, first part we give sequence and structure information. So, for example, if you have the data, right, first we need to see which protein. In this case we give the protein name like ribonuclease HI. So, obtain from E. coli, because source is also important. Sometimes we take T4 lysozyme, and the lysozyme we can also from the chicken or human, then the sequences are different, right. So, in this case it is very important to provide the source information. So, we give the name and the source, right. When you give this information, you can also get the length and the molecular weight, and give the codes where we can get more information for the particular protein. So, we give the PIR sysprot, and currently we give the uniprot ID, and this is the enzyme, then we can give the enzyme number, and we give the link to Brenda. What is Brenda?
Enzyme database.
Enzyme database, comprehensive enzyme database, right. So, if you a particular protein is an enzyme, then you can give the link. So, you can get more information if anybody is interested to know about the particular enzyme. Then PMD number is the protein mutant database, right. They give the literature information, right, regarding the particular mutant, and then we give the PDB information. This is the wild type, and if the mutant, the structure is known, mainly for the case of lysozyme, in that case most of the mutants the structure is known, then we give the data the PDB mutant. If it is not known, then it is blank. Then we give the homologous PDB entries. What is homologous PDB entries?
Proteins.
You can see the proteins which similar to the wild type structure. For example, with 2RN2, you can see the proteins which are similar to 2RN2. So, there you will get as the homologous PDB entries. Then we give the mutation information, right. For example, here K91R. What is the meaning of this K91R? Here lysine
At position 91.
Position number 91, this is the position,
Mutated to arginine.
And this is mutated to arginine. Here they did the analysis because lysine and arginine both are positive charge, they want to see the length, right, what will happen. So, this way they changed the lysine to arginine. Then we give secondary structure, right. Here this is in coil is coil, right. If you want to see the PDB in the cartoon, right, you can click here. So, you can see the cartoon structure and locate the mutant, you can see this in coil structure. What are the other regular secondary structures? Helix or strand. So, here it is in coil region, right. So, accessible surface area, so this we discussed earlier. So, you can see how far a residue is accessible to a solvent. So, this is 80.5 angstrom square, this buried or exposed, it is partially exposed, right, because it is more than 80, right, more than 50, so you can see the partially exposed or the exposed region. So, if you have a particular mutation, we give all the information regarding the protein and the mutants. Then we give the link, okay, this is the KH91 to R. So, we display the mutation, you can see the lysine 91, okay, here is the lysine 91, right, K91, and see the residues which are occurring near to this particular mutant. So, if you see the yellow one, okay, this one, right, here this is about within 0 to 4 angstrom, right, this is 0 to 4 angstrom, and the green one, so you can see this is 4 to 8 angstrom. Then here I display the data here, 0 to 4 is 88 arginine, 89 glycine and other residues, if you see the side chains here, and the if you see this 4 to 8, this is the green one. So, here also you can see the residues, right, which are occurring within 4 to 8 angstrom. For example, green, this is a tryptophan, you can see a tryptophan 90 here, this is 2 rings here. Here also you can see the tryptophan here, right, you can see tryptophan here, right, in the 4 to 8 angstrom. So, we give the data, we give the picture representation as well as give the data, you can see which are the residues which are surrounded by the any particular mutant residue. So, we give the experimental protein information right from the uniprot and the PDB as well as the mutant. Now, we give the conditions, what are the experimental conditions they use to get the data. The important one is temperature, right, and the pH and the buffer name and concentration and the protein concentration and different measures, whether they use CD or DSC or fluorescence. Here they use CD, and which method they use, thermal or the denaturant, here they use the thermal. So, what are the information which you can obtain in the literature, whether they report in the literature when they do the experiments, then we give all the experimental conditions. Now we get the data, whether you get the data by denaturation, right, for example delta G H2O for the wild type, this for the mutant, right. When we extrapolate the data, we can get the slope and the midpoint of the concentration, we will get the M and Cm. Likewise, if you get thermal denaturation, you can get the Tm or the delta Tm, and still delta G, right. You can, if you know the heat capacity and any particular temperature, you can get the delta G. Then the reversibility, right. What is the meaning of reversibility? If you folded state, unfolded state, and other conditions, you can come back, right, from the unfolded state to folded state. That is called a reversible. And then the states, that how many states, for example unfolded to folded, or they are having an intermediate states. So this will give you the state. If it is 2, then it is having a folded state and the unfolded state, then give the activity of that particular protein. So if you see the data, so here delta G H2O is 6.4 and delta G is minus 3.1 kilocalorie per mole. That means it destabilizes the protein up to 3 kilocalorie per mole, because it is delta G H2O. Then we have the thermodynamic data, we can give that Tm is 52 and dTm is 0.1, and give the enthalpy values and the because no M and Cm because thermal denaturation. If it is denaturant, we give all these values. So reversibility it is yes, right, because it is completely this protein is reversible protein. Then we go with the literature, right. So we have the protein information and we have the mutant information and we have the experimental data and the experimental conditions, right. Now we link with the literature. So here we give the keywords that are used in this paper and the original reference where we get the data, right. For example, here this is JBC, right, published in this one, and we give the link to the PubMed, this literature database. From that you can get the free text for most of the journals. So you can obtain the papers, and if you want more information, it is possible to get the information. Then here we add one more aspect, that is related entries. So this means, so for any particular mutant or particular delta G values, we gave the data, right, what are the entries which contain similar protein and similar type of mutants. So these are the entries which deal with the RNase H and the different type of sources, for example this E. coli. So you can see all these related entries and you can compare what are the different conditions, what are different papers, and how they got the different types of data and what influences the stability, right, based on experimental information. So give all the information and we look at the statistics, right, here totally about more than 25,000 data. So this is good enough to carry out any analysis, and if you see this one, they obtained from 700 proteins, more than 700 proteins, and from about 2000 papers, right. The data are obtained from different proteins, about 1800 proteins, and you look at type of mutations. This is dominated with the single mutation, this is about 12,000 single mutants and some double mutations and multiple mutations. For the wild type also we have a lot of data, about 10,000 wild type data. So currently if it is PDB, there are about 130,000 structures, and wild type 10,000 data, but they are redundant. Sometimes you can see the same protein at different concentrations or different experimental conditions, right. And in this case, if you take the non-redundant ones, you will get a less number of data, but that you can do for analyzing these proteins to understand the stability with respect to this contribution of these various interactions. So we look at the secondary structures. So you can see the most of them are helix followed by strand, and also we have sufficient number of mutations in the turn and coil. This you can use this information to understand the factors which influence the stability in helix and strand, whether the same or different. You can see, you can carry out this analysis. In the accessibility, about 6,000 buried and 4,000 in the partially buried and exposed regions, right. So that also you can see the importance of the location of the residues, right, for mutations. Then about 15,000 thermal and the 9,600 obtained from the denaturation experiments. So now we see the mutations, right. For example, if alanine is mutated to valine, alanine mutated to glycine. So we showing the frequency of mutations, right. Here totally how many mutations possible?
20 into 19.
20 into
19.
19, this is equal to 380 mutations, right. Now here this is from, this is 2. So for glycine is mutated to alanine, glycine to valine, if this means glycine to valine. So among the 380 possible mutations, If you look into this table, some mutations they study widely studied and some mutants very rarely studied, some cases they are not studied. For example, what are the mutants which are widely studied? Yeah, mainly some of the residues to alanine, so you can see shown in red is more than 100. See valine to alanine, isoleucine to alanine, right, and methionine to alanine, because mainly because of these alanine scanning mutagenesis studies, they can get the data for the mutagen to alanine. This is the reason why we get more number of alanine mutations. And likewise, other than that, you can see several other mutations, mainly similar type of residues. For example, isoleucine to valine 430 times, like valine to phenylalanine, you can see the several types of mutations, similar type of mutations. And also you can here you can see the asparagine to aspartic acid and glutamine to glutamic acid, lysine to glutamic acid. Why they study lysine to glutamic acid?
Destabilizing.
To see the negative charge to positive charge, what will happen if you mutate whether this is stabilized or destabilized, right. So, here also some cases there is completely 0, because they want to mutate tryptophan to proline. Likewise, you can see the other mutations, methionine to tryptophan or methionine to these residues. So, initially we started to analyze the different mutants, then some of the cases is really we know that they should stabilize or destabilize, right, and interior core they do not want to mutate alanine to aspartic acid. So, in this case they will destabilize, there is no fun to do it again, right. So, they want to know to some cases if it is very curious, this is the reason they have some mutations to some specific mutants, and some cases it is very rarely occurring. Okay, now how to get the data? For example, if you have a lot of data and if you are interested to get any specific data, right, we have two different types of search options. One is the basic search or one is advanced search. The basic search, it will search from all entries in the data, especially the keywords and other specific entries. So, if you give the keyword or the protein name or any source name or anything, just if for example, you put lysozyme and click on go, then this will show you that all the data which contains the term lysozyme, right. So, all the proteins lysozyme and you will get lot of data, right. In this case you will get almost all the information, right. Then the advanced search, we have given different options. You can search with the protein name, you can search with source, you can search with the mutation. For example, if you are interested only in single mutation, then you can click on single mutation. Then if you want to see the additivity effect of double mutation or multiple mutations, for example, if you have a double mutation, right, A25V and R72K, whether the stability obtained with this mutant is a sum of A25V plus R72K, we do not know. So, if you want to do this analysis, you get the double mutant and get the single mutants, right, and add up these two and see how far you can relate, right. It is related, you can relate good correlation or less correlation. If yes, why it is yes, if no, why it is no, right. So, if you want to do analysis, you can do double mutation, the multiple mutations more than 3, right, and then see how the delta G value changes. Also in several other aspects, for example, thermophilic proteins, if you mutate few residues, it enhances stability more than 10 to 15 kilocalories. If you see the multiple mutations, right, and see few mutations, if increases stability by 10 kilocalorie per mole, you can try to understand why this is very high. Is it possible to introduce this type of mutations to some other proteins that will also increase the stability, right. In that case, you can get the proteins with the high temperatures, you can withstand high temperatures, that have several biotechnology applications. This will get the data for single mutation, double mutation and multiple mutation. Then you can try to get the data based on different secondary structures, for example, helix or strand or turn or coil. Then accessibility. So, here two options we have. One is we can get from the buried or partially buried or exposed, the data we have already, or we can give any range in percentage, or you can also do it for the Angstrom square. Angstrom square is the real value we obtain from the program, and percentage is normalized with the extended cell accessibility, right, you can do that. Then measure and the method, pH you can define, okay, because neutral pH, so I put 5 to 9. Then the delta Tm or Tm, for example, if you want to identify only the stabilizing mutant, in this case you put delta Tm or delta G H2O or delta G which should destabilize, then should be minus values. If you do like this, you can get the stabilizing structures. Or sometimes if you want to identify the mutants which have extreme stability, right, not around 0. In this case you can see that is minus 100 to minus 2 and here to plus 2 to plus 100, right. Then you will get the stabilizing and destabilizing extreme values and see what is the difference. Or if you want to see these minor changes, what are the mutants which do not make much changes around 0. So, minus 0.5 to plus 0.5 you get the mutations and then see how this is attributed with no change in the stability, right. All these things if you do these numbers and you can get all the values. Energy values you can give either kilocal or kilojoule. So, both are possible in this database. Then state you can see 2 state or 3 states or more than 3 states. Reversibility also you can define, or also you can search with the keywords and others, and then if you want the current data you can put the year, you change the year, right, you will give the data which are reported recently in the literature. So, these are the various secondary structures, right, and accessibility that we discussed earlier. So, on this aspect you can search this particular database. Then you can display options. I put all the terms which we use in the database, but if you display all the results it will be big mess. So, we give options to the users, they can choose. For example, if they use thermal denaturation, there is no point to click on the M, Cm and delta G H2O because we do not have any data. If we denaturation there will be no data on Tm. So, accordingly what they require, they can click on the terms and they will get the data. It is also possible to sort the data based on the wild type residue or the mutant residue, or you can see the residue number or the depending on delta G values, right. We have 4 different sorting options, you can use all sorting options. It will give the priority, this is a priority 1, priority 2 and priority 3 and priority 4. For example, if you sort by wild type residue, mutant residue, alanine to valine, there are many alanine to valine. So, they display from the beginning. So, then if you third, if you use a mutant number, then show the first, the number least number at the first. If you want to have this mutation with the highest delta G values, then if you start with that one, then all the alanine to valine you can see at the first place with the highest delta G or delta Tm. So, you can also do it by ascending and descending, any order you can use it, right, and then you can hit is default is 300, but you can increase it to get all the data, right, at one stretch. So, now you have the results. So, if you based on your conditions finally you get the results, and it is also possible, right, to download the data, right, for the analysis. So, when you download the data, then what are the potential applications, how can we apply this Protham data, right. The Protham has several applications. You can say first one, you can see you can estimate the stability of protein structures that we discussed in the previous class, right, because delta G experimentally is known. So, you can do the contributions and you can relate, right, and here we discussed about the mutation. So, we can try to understand the factors which influence the stability upon mutation, either different secondary structure or different locations, right, that you can understand. Then if you have any data, if you predict any data, you can compare the experimental data because experimental data is available, right. And the third one, we discussed about stabilizing residues. We can also try to compare the residues identified as stabilizing or also experimentally obtained from these experiments, right, CD or the DSC. Then you can develop models for predicting the stability about mutation, right, whether the single mutation or the double mutation or multiple mutations, right. So, you can use various potentials or various features what we relate the stability with the properties, and we can use these properties to develop models. Then you can check the additivity. For example, if you have the multiple mutations 3-4 mutations, right, whether the single mutations add up together, this is similar to the multiple mutations or totally different, how can we account that, right. Then you can see the extreme stability mainly for thermophilic proteins, right. If you make few mutations whether the stability is increased, right, how to account that stability. Then see the mutants which can change the stability at least more than 2 kilocalorie per mole, either destabilize or stabilize. So, you can see these mutations and analyze what factors for the extreme stability, or also you can see the narrow range of stability, right, there is not much change, right. So, then we can design the proteins with enhanced stability. For example, if you know the some mutations which are important to enhance the stability, then try to utilize this type of mutations, right, and then make a protein, design a protein with enhanced stability, it can stand at more temperatures, right. The potential applications in biotechnology. So, like as Pratham is very useful, right. This way we get more than 1000 citations for this one, it is continuously used by the researchers, right. So, you can also use this, right, for any of your projects or your any of the your research. Fine. So, till now what did we discuss today?
Stabilizing residues.
Right, the first part we discussed about stabilizing residues, right. What are the various factors or properties considered for identifying the stabilizing residues? Hydrophobic character, long range interactions and confidence score. Based on these characters features, we derived 4 parameters, right, surrounding hydrophobicity, long range order, stabilization center and confidence score. So, we get the data for all the residues in protein structures and we make a cutoff, right. Based on the cutoff you can identify the stabilizing residues, and we analyzed, okay, these stabilizing residues have some specific properties, right, and compared with the B factors, right, and as well as some of the experimentally known information. In the second part we discussed about the database. What is the name of the database? Protham, right. This is thermodynamic database for proteins and mutants. This is why they put the name Protham. So, this is thermodynamic database for proteins and mutants, right. So, if you see the Protham, I show this figure. So, here this is a folded state, okay, this is unfolded state and this is a curve. Yesterday I showed about the thermal denaturation curve, right. This is how we designed this logo to show the thermodynamic stability for proteins and mutants, right. What the information we have in Protham database? Experimental data, mainly the thermal stability or the stability upon the denaturation delta G H2O, melting temperature, and supported with the experimental conditions, which conditions they used, right, to obtain the data. Then we have additional information regarding the sequence and the structure and the mutation, right, as well as the literature information, right. Then we have different options to search the data as well as to obtain the data and sort the data. Once you get the results, then it is able to download and you can use the data for any of these potential applications, because Protham has several applications. In the next class I will discuss one of the applications, how to relate different amino acid properties to the stability of the mutations and the models to predict the stability upon mutations. Thanks for your kind attention.